Hi friends, welcome to Botany, the language of plants, and today's topic Anther development. We know that flower contain androecium and gynoecium. Androecium or stamen contain anther and filament, and gynoecium contain stigma, style, and ovary. We know that anther contain pollen grains. So these are pollen grains. After dehiscence of anther wall, these pollen grains come to stigma and fertilization occur. So this is the basic concept about fertilization, and it is a part of embryology. Now, at first, anther development. How anther developed? Anther development occur in two steps. In first step, anther developed its morphological shape. So, then pollen grains produce through mycospore mother cell. And then in second step, dehiscence of pollen grains occur. So, in this video, I will discuss about first step, that is anther develops its morphological shape, and then pollen grain produced through mycospore mother cells. So, at first, stamen developed from stamen primordia. So this is floral meristem. Now, sepal develop from here, then petal, then stamen primordia. This is called stamen primordia, and this is carpel. So carpel produced from here. Now, so stamen primordia always originated from third hole. This is first sepal, then petal, and then third number stamen primordia. Okay. So from outside of a floral meristem, first number in first hole present sepal, second hole present petal, and in third number stamen. Now, some genes like APETALA3 or AP3, pistillata, or PI gene, agamus, or AG gene work to produce stamen in case of Arabidopsis, and plena gene or PLE gene found in snapdragon. So these genes are responsible for stamen development. Now, so we know that androecium developed from anther primordium. Anther primordium contain three layers. From first layer, epidermis produce. From second layer, archisporial cell, anther wall, and microspore mother cell produce. And from layer 3 or third layer, connective tissue produce. That means xylem and phloem. Now, gradually, anther primordia change its shape, contain four-lobed, and hypodermal cell of each lobe became differentiated by their shape, contain a prominent nucleus and dense protoplasm. These are called archisporium cell. So layer 2 produce archisporium cell. Now archisporium cell divide into outer primary parietal cell wall and inner primary sporogenous cell layer. So from archisporium cells, outer parietal layer and primary sporogenous cell layer produce. Next, anther lobed became more round in shaped. Parietal layer divide and produce endothecium and secondary parietal layer. So from outer parietal layer or primary parietal layer, endothecium and secondary parietal layer produce. After producing secondary parietal layer, it divide and produce middle layer and tapetum. So middle layer and tapetum. So secondary parietal layer produce middle layer and tapetum. Now endodermis, middle layer and tapetum together called anther wall. When outer parietal layer produce anther wall, at that time simultaneously primary sporogenous cell layer divide and produce spore mother cell. So spore mother cell present. So these are spore mother cell. From primary sporogenous cell, this spore mother cell produce. Now these spore mother cells divide and produce pollen grains. So spore mother cell divide by two types, simultaneous type division and another is successive division. Simultaneous division found in monocotyledon and successive type division found in dicotyledon. So in case of simultaneous division, after meiosis, spore mother cell produce two nucleus within a cell and then by second meiosis division produce four nucleus. So in this case meiosis occur, cell division occur, but partition layer absent. So this type cell division called simultaneous type division. Now at the final stage, nuclei separated by layer and produce spore tetrad. In case of successive type division, first meiosis occur and spore's mother cell produce two nuclei which separate from each other by a cell wall. So this is called dyad. It's not a cell wall, a layer by a layer. So in case simultaneous division, dyad not form, but in case of successive type division, dyad form or dyad produce. Now second meiosis occur and tetrad produce. Finally these tetrads by separation pollen grains produce. So this process from microspore mother cells to produce pollen grains is called microsporogenesis. So this process is called microsporogenesis, and this pollen grain stay in this portion. So this is pollen sac and here pollen grains are stay, and here connective tissue present. So this is a mature anther where epidermis, endodermis, middle layer and tapetum present, and pollen grains present and within this pollen sac. So this is all about anther development. If you like this video, then press the like button, press the bell icon to get notification, subscribe to my channel, and thank you for watching this video.